and reduce the textile dyes, reduce the usage of textile dyes while keeping our textile industry intact. So that is the objective of this study. But what is the issue of using synthetic dye versus natural dye? Synthetic dyes are readily available. In a synthetic lab, you can just easily produce them. They are efficient, cost effective. So if you order one type of dye, you can get the exact same dye, reproducibility and wide range of colors. What's the issue of using natural dyes? These doesn't have these positive benefits. Like they are not easily accessible, they are not cost effective, and worst thing is they are not reproducible, and we can't get a wide range of colors using natural dyes. But the positive feedback is about the natural dye the consumer prefer because of the carbon dioxide footprinting, eco-friendly, biodegradable, and soothing shades. But if we want to go into a commercial scale, these we cannot go with the available positive factors. We have to break through the big barriers of reproducibility, cost effectiveness, and reproducible and easily accessible. So challenges what any industry faces in natural dyeing is availability. For example, the promenade peel gives a very good natural dye. But where do we get the promenade peels every time to form it into a natural dye? Reproducibility further. Let's say the promenade peel taken in month of January and taken in month of December or in June will differ. It will differ from place to place, country to country, time to time. So how do we get exactly the same dye if we are going with a natural source? And further is the cost to extract it from natural resource. It is cost. So we can't compete with the readily available synthetic dyes, which is just put into a factory and produced. So how do we overcome this? Just I want to brief you a little bit about the Sri Lankan tea industry. Sri Lanka exports around 300,000 metric tons of tea per annum. So the good quality, the first leaf, second leaf, and the third leaf of the tea is only exported as high quality tea. So during this high quality tea export, there are 1/10 of that tea is removed as broken tea or broken mixed fannings, which is like broken pieces of the good quality tea. So 1/3 is like 30,000 metric tons of this waste tea is generated during tea exports. So what currently Sri Lanka does is it uses partly this broken mix fanning, that is the broken pieces which is generated by selling of the good quality, and extracts instant tea, which is something like Nescafe. Like Nescafe, you put it into water and it's completely soluble. So they extract instant tea and sell it to the world market. And actually, Sri Lanka supplies 70% of world's supply of instant tea. So during this instant tea manufacturing process, there is a huge amount of waste generated. That means from broken mix fanning, when we form instant tea, there is a huge amount of waste generated, which we have used to synthesize our tea dye to dye the fabric, which is commercialized under the name tea hues. So basically, Unilever Sri Lanka is the largest supplier of instant tea, accounting to 50% of global supply. and functions as a strategic unit for the Lipton iced tea supply chain and supplying over 70% of the ready-to-drink volume sourced by PepsiCo Lipton International. So whatever the iced tea bottles sold by PepsiCo International, 70% of that instant tea powder goes from Sri Lanka. So during this process, there is a huge amount of waste generated. Just imagine around 20 metric tons a day. So the objective of the study was to use this waste as the possibility of using this waste as tea dye. So the reason for sustainability of this process is because now we don't have to go and start extracting or collecting the tea waste and extracting tea dye because the continuous supply chain is already there. One company is the global supplier. Sri Lanka is the global supplier. The supply chain already exists. What we need to do is formulate a method to use it as a tea dye, the waste of that as a tea dye. Uniformity. The other problem is, as I told you, the promignate peel changes from time to time. But as we are supplying instant tea of constant grade, of constant quality, constant flavor, constant taste, constant color. So to supply that instant tea, we are using the same quality tea. So if we are using the same quality, we will end up with the same product every time. And this can be converted into uh, easily usable synthetic dye powder. And other advantages, by using something from waste, we will be reducing the carbon dioxide footprinting. So the SLINTEC, which is the base, the research organization where I was attached, and which is the research arm of this project, did a partnership with the Unilevers and the Dynawash, the dying people, and we did the research part and found out the method how it should be done. And using these two partners, we came up with the manufacturing process to use this broken mix fanning, that is the tips broken off while we are exporting the good quality from Sri Lanka, the waste tea, how we can formulate into a tea dye. And we have a local and an international patent on that. And now I'm going to share with you a video clip to show you you can see the biggest issue of using natural dyes. You can't obtain the fastness properties like the color wash fastness, the light fastness. But using this technology, we have been able to obtain the required fastness properties utilizing this natural dye, and currently it is marketed in the world under these branded um, brands such as Pink, Levi's, Aritzia, Athleta, Mark and Spencer, Club Monaco, and many more. And thank you for your kind attention, and I'm open for any.
Okay, thank you, Doctor, for your nice presentation. Please, any question you can ask Dr. Benita.
Could you please leave questions at the end?
Okay, so now move to.
Okay.
Okay, so now move to the next presentation. Okay, so the second presenter is with us, Dr. Rajani Sirvistava from the Environmental Sustainable Development Department, Rajavi Gandhi South Campus, Banaras Hindu University from India. And she will present us the porcelain, Portulaca oleracea, a prospective source from the national nutritional security and development developing countries. Now you can start, Dr. Rajani.
Yeah, good morning, everybody, from panelists, and good morning, Baghdad. Myself, Dr. Rajni Shrivastava. Presently, I am going to present on the topic Purslane, that is Portulaca oleracea, a prospective source for nutritional security in developing countries. For that, I am going to share my slides with you. Is it visible to all?
Okay, it's on the screen already, Doctor. Can start.
Yes.
Yeah.
Should I start? Okay. Yeah, so my topic is Purslane, a perspective source of nutritional security in developing countries. Uh, Myself, Dr. Rajni Shrivastava, working as assistant professor in Banaras Hindu University, Varanasi. So, as we know, food security and natural resource management is the today's major challenge, especially in developing countries like India, where population growing is exponentially. And interesting thing is that about 80% of the world population is live in developing countries, and it is assumed that in about 2050, the share will increase further to 90%. So, the modern agricultural practices like the green agricultural practices and many other things has been suggested for this, but this only marginally increase the food production. However, the evidence indicate that the changing climate may adversely affect the productivity. Therefore, if food production would not be enough to meet the dietary demand of ever increasing population, there may be shortage of food supply. So only one option before us to move backward toward our traditional knowledge. So sustainable food production through integrating knowledge of indigenous communities, or integrating knowledge of indigenous or traditional knowledge and local communities, has significant for meeting the UN SDGs, the different UN SDGs like no poverty, zero hunger, good health and well-being. So and so I have chosen a species, well-known species, that is the Partulica oleracea, well-known by local community, not at global level, and it is commonly called purslane species, having lots of nutritional, medicinal, pharmacological, and phytoremediational properties and significant. And so the aim of my study is to study the ecology, distribution, characteristics, and benefit of this plant, as it uncovered immense nursing potential and has grown as the one of the important herb for future, especially in climate change scenario. Now come on the ecology of purslane. This plant purslane, that is called Portulaca oleracea, is a succulent summer annual plant and cosmopolitan in distribution, as it is among the eighth most common weed of the world, eighth most common plant of the world, herb of the world, and it occur everywhere. Means it can occur in riverine, riverbed, garden, as well as in dump site. So the name Portulaca is derived from a Latin word, that is the Porto. Porto means milky, since the plant contain a milky juice. And the binomial nomenclature, like the Portulica oleracea, the Portulica means little entryway, and it may be little entryway because of the kind of dehiscence of the natural products, while oleracea means vegetable, showing the diffuse utilization of this plant as food from old time. So this plant is recorded as one of the most valuable restorative plant and named global panacea by the World Health Organization. And this plant is belong to the family Portulaceae and the order Caryophyllales, class Magnolopsida and Magnolophyida division. In this slide, you can see the general habit and habitat of this purslane plant. You can see here the general habitat of the purslane plant, little matty type of a root growth it have. And this second diagram shows different stages of the plant, and it has tender stem, light pinkish color stem it have. And this is the succulent leaf, and this diagram shows a bunch of capsule, stems bearing bunch of capsules. And this is the structure of capsule after releasing the flower. And this is diagram of flower. And this contain capsule, seed containing capsule. And it has, Portulica has black colored, dark black color small seeds. So this is the general appearance, or we can say habit and habitat of the purslane. In terms of its distribution, regional and global distribution, there is little uncertainty about the origin of purslane, but it is increasingly present in the Mediterranean region for the most part in the dry and semi-dry land of the northern Africa and South Europe, especially in Saudi Arabia, United Arab Emirates, and it has been reported as a native to Europe, Africa, North America, Australia, and Asia. In India, it is distributed in Andhra Pradesh, Arunachal Pradesh, here, Bihar, this area, Chhattisgarh, Chandigarh, Gujarat, Goa, Himachal Pradesh, Uttar Pradesh, Uttarakhand, and here in Mizoram, Manipur, and Nagaland. So it has broad regional as well as global distribution. In terms of nutrient profile, purslane leaves and stems can be used in raw or cooked form. Both form we can use purslane. Its leaves can be used, its stem can be used. We can use its plant in raw form as well as in cooked form. It have it is highly nutritious and having all essential minerals and proteins and vitamins. The purslane species you can see here have maximum amount of water content, approximately 93% water content it have, and very less carbohydrate, 3% carbohydrate it have. and high protein, 2% protein content present in the purslane. It contains dietary minerals like you can see here in this chart, dietary minerals like the potassium, magnesium, phosphorus, sodium, and calcium. And the potassium content, it have greater potassium content, approximately 494 milligram of potassium present per 100 gram fresh weight of the plant. And this potassium is very much useful because it helpful in the reduction of hypertension, and it helpful in the prevention of the bone diseases like osteoporosis and kidney injuries. This plant contains the maximum amount of vitamins among leafy green vegetables, followed by other vitamins like vitamin C and other. Only vitamin K are absent in this purslane plant. It contains thymine, you can see here, thymine, riboflavin, niacin, vitamin B6, pantothenic acid, and vitamin E. Alpha tocopherol has a significant role in many metabolic procedures like cell signaling, regulation of gene expression, and immune functioning. Lipid and fatty acid composition differed in different part of plant, like it differed in leaf, stem, as well as in flower. So in case of fatty acid, saturated fatty acids are higher in stem plant. Among all, the most abundant form of fatty acid is polyunsaturated fatty acid. Approximately, you can see here is 78 to 79% polyunsaturated fatty acid present in the leaves, and approximately 72% in stem, and 62% in the flower. So, among all, the most abundant form of fatty acid is polyunsaturated fatty acids present. There are four different type of omega-3 fatty acids found in the purslane, like alpha linolenic acid, 11 and 11 carbon essential omega-3 fatty acids, and palmitic acid, oleic acid, as well as fatty acids. As we know, alpha linolic acid is required for normal growth, and our body don't produce omega-3 unsaturated fats. Hence, omega-3 unsaturated fats must be consumed from dietary source. So for this, purslane species is a good example. Besides nutritional and medicinal uses, purslane also have many benefits. You can see here in this diagram. It acts as a relaxant. It is found that juice or extract of this plant contains a high amount of potassium. It acts a potassium ion, which help in the relaxation of the muscles. It has anti-inflammatory benefits. Oleracone and alkaloid found in the purslane, which showed an anti-inflammatory effects. And various results confirm that aqueous extract of purslane may have a potential therapeutic impact by representing the vascular aggravation process in different vascular diseases like atherosclerosis. So it has anti-cancerous properties, anti-ulcer properties, and the extract of purslane has also proved to be useful against the growth of hyphae of genera trichophyton. Means it has antifungal effect. Root extract of the purslane has been reported to inhibit zoospores motility without killing or busting the zoospores. So purslane can used in pest control, and this plants also shows phytoremediation properties. In this manner, the huge scope of purslane can be advantageous for accomplishing the food and healthful security and different advantages at local and regional level, allowing a chance to preserve agrobiodiversity for sustainable development practices. It is very simple in terms of cultivation because it can be spread through vegetative propagation or shoots in wet soils. It produces an enormous amount of seeds. As you have seen in my slide, that there are black color small seeds produce in enormous quantity. So this plant can be propagate through vegetative, through seeds. So it has various mode of reproduction, so we can easily cultivate this plant. These plants are adaptable to moist soil, including mildly saline soil, and it has drought tolerance capacity. It was observed that stem cutting with with nodes has 70% survival, and with internodes has 0% survival. That means if we wish to cultivate this plant, we have to cut its stem with nodes, so the chances of survival will be more. It can be grown at elevation up to 1700 meter in the tropics, and it can grow best at the temperature ranges 18 to 32-degree centigrade. The plant is effortlessly spread by running water. And the most important thing, that this plant take around 6 to 8 weeks to create a harvest from a seed. So we can conclude that purslane species are highly beneficial and can be easily cultivated in tropical, subtropical, and temperate region of the world. In India and other part of the Asia, this species is luxuriously growing and found in almost all agroecological zones in the country. As this plant doesn't require a lot of agronomic expertise and can be easily grown compared to other nutritive plant, will be an ideal substitute for resource poor agriculturists in developing areas. Besides this kind of local species cultivation practices will set a model before local farmers for conserving other discarded and underutilized crop in different agroclimatic region of the world. So this is all about my presentation. Thank you so much.
Okay, thank you very much,
Thank you,
Doctor,
sir.
for your presentation. Now we have another presentation presenter with us, Dr. Hashim Saeed Murad from the Department of Agriculture Extensions and Rural Development on the College of Agriculture Engineering Science University. Now present us the level of economics participation of rural women in the whole governorate Kurdistan region of Iraq. Now you can start, Doctor.
Morning, everybody. At the beginning, I want to award my thanks to order. I am Dr. Hashim, Assistant Pro from the College of Agriculture, Engineering, Science, Agriculture Extension, and Rural Development. The level of economic participation of rural women in the whole governorate in Kurdistan region, Iraq, and its relation with some variables. Forward statement. In a semi-desolated northern part of Iraq, Kurdish women are striving to extend their role in a very conservative society. The authorities have opened a little hope for those women to retain their extensive by providing them with limited training to enhancing farming activity to show their abilities. This research has proven that Kurdish women are up to the level of trust. This map for Iraq and the whole governorate have many districts like Zakro, center of Dahod, Amedi, Semil, Shekhan, Akre, Bardarash. And this picture clear many activities for rural women. Introduction. Society development depends on combination of two basic aspects, the people and the government efforts by means of which social, economic, cultural, and physiological change are interrelated with an eye to improve and upgrade the status of the people of these communities. Women play crucial roles in the agriculture production in the developing countries, especially in countries with low income rate and where agriculture account for high ratio of the gross national production. Rural women represented the majority of the agricultural labor force in these countries because they produce the bulk of the locally consumed foods. This qualified them to be one of the fundamental variables in achieving the economic development and family welfare in the rural communities. Curbing all the agriculture potential constitutes a contributing factor in the slowness of development and absence of food security. The outcome and recommendation of many conferences and studies highlight the need to pay attention to women and their participation in different aspects of an in order to achieve a true development. In view of the considerable participation of rural women in the affairs of the local community and in the light of economic significance. Rural women, as half of the rural population, have an important roles in economic and social activity, and to achieve rural development requires more attention to women that are involved alongside and tandem with men in economic and social activity in expanding the rural woman importance role in production. This, thus we know that the achievement of the World Conference on the United Nations the Decade of Women, which is entitled Equality, Development and Peace in 1980 was held in New York, was that two-thirds of the total work done by women in the world, which rural women do most of this work. Therefore, if the overlooked women in the production and does not matter to their importance role in development and income status, each types of planning will fail and always lags behind the country in general and the village in particular, or in other words, their lack in field of different agricultural activity not only makes trouble. The family in the area of function that are responsibility as a mother and wife, but is serious as power efficient and effective in terms of rural economic caused a crisis. Women make essential contribution to the agriculture and rural economic in all developing countries. Their roles vary considerably between and within region and are changing rapidly in many parts of the world, where economic and social forces are transforming the agriculture sector. Objective of the study. The main objective of the study is to determine the economic participation of rural women in the whole governorate. The specific objectives are to identify the economic participation level by rural women in the whole governorate, to identify the relationship between selected characteristics of rural women and their degree of participation in economic activity, to diagnose the obstacle of economic participation by rural women in the whole governorate. Why is sustainable agriculture so important for developing countries and the rural poor, because 63% of population live in rural area, 73% of poor live in rural area. Agriculture and agro-processing account for 13 to 60% of GDP in developing countries and an even large share of employment. Even with rapid urbanization, more than 15% of the poor will be in rural area by 2035 and depending significantly on agriculture. Methodology. Total of 474 respondents were randomly selected for different rural areas of the whole governorate. One is interview based in structured questionnaire from DOC, which has been held from April to June 2015. Their Likert scale was used to identify the level of participation in economic activity of alpha value obtaining for the scales was 0.750 exceeding. The collected data was subjected to data analysis, including statistical such as frequency, percentage mean, standard deviation, correlation, and regression using SPSS software. The table shows sample size of study. Center of the Hook, number of women above 18 years of age, 505, size of sample, 50, Semel, 67, Zako, 40, Ahmedi, 55, Shekan, 47, Akre, 64, Bardarash, 151, is total 474. Result and discussion. Identify the economic participation level by rural women in the whole governorate, the whole Kurdistan region of Iraq. Table 2 show the rural women in the whole governorate are divided into 3 categories from table. It reveals that 30.169%, 143 of respondents have high level of economic participation. The table also shows that for 47 or 257 percentage, 200 to 24 have moderate level of economic participation, and only 107 of the respondents have low level of economic participation. Maybe the reason is that women participation in agriculture. According to Table 3, items of respondent participation in economic work were ranked based on their mean values. Respondent participation level toward the item assured to cut home expenses is ranked the highest mean value, which was to 592. This may be due to difficult economic condition experienced by families in the region, especially the 1990 economic blockade, and therefore rural women seek to reduce the expenses of, of the house and provided them in anticipation of similar circumstance with such item as I, I present to the official proposal for development project for the region, ranking the lowest mean value 1 of 582. This means the most of the respondents have not faced problem with financial. Identify the relationship between selected characteristics of rural women and their degree of participation in economic activities. Person and sperm correlation were used to examine the association between independent variables and degree of economic participation of respondents based on the result in table 4. There is significant correlation between each of age, number of study years, classification with service in the village, urban openness with the economic participation of respondent in study area. Age has a positive significant relationship with the economic participation of respondents. The number of person correlation value 0.132. This was significant at at 0.101. The reason may be due to accumulated experience and increasing awareness of the importance of participation in economic activity to improve their position with the family and society, followed by number of years of study has negative correlation with the economic participation, person correlation value 0.210. This was significant. Maybe reason is the majority of rural women in the target area, the illiterate or fall into the low level of education. Other characteristic is classification with service in the village. The results show that there is a positive correlation with economic participation. The number of person correlation that there is positive correlation with economic participation. The number of person correlation value or 0.155. This was significant. Maybe there is for that where respondent has high classification with service in the village is will encourage them to economic participation and of course benefit from variable economic opportunity in this village. To diagnose the obstacle of economic participation by rural women in the governorate. Table 6 shows the obstacle facing economic participation of rural women, that the obstacle high unemployed rates is ranked first with an arithmetic mean 2.713, and that of women economic dependency occupied the last rank with an arithmetic mean 2.156. In brief, the economic activity is one of the most important factors which can increase the ability of rural women by giving them new courses or new training to make them more able to indicated that the majority of rural women in the governorate that 13 percentage 143 of the respondents have high degree of economic participation level. This study result also reveals that the greatest variable hindering rural women participation in the targeted area, which are between age, number of studies, classification with service in the village, urban openness based on the result and observation made during the survey. It was clear that the participation of women in most areas is still less than the expectation based on the result of the study. Conclusion and recommendation. The following recommendations were made. Agriculture extension agents in the target area should encourage and cooperative the rural women to participate and be member of civil society institutions and NGOs or CBOs, community-based organization. The extension agents should have a reorientation of the program and activity where women will be treated equally with their male counterparts. This will ensure more rural and agricultural development in the target area. Create an environment that encourages rural women, increases participation in extension household decision making. Awareness raising program about the importance of rural women participation should be made both for males and females, religions and rural leaders, women, associations, civil societies, NGOs, and mass
Dr.
media
Said,
can play.
excuse me, if you can hurry up because you have limited time to present.
Yeah,
Yeah,
the
I'm so
last,
sorry about it. Yeah.
develop and promote personal skills training course for rural women in leadership, public speaking, family planning, decision making, and self assertion. Expectation: it is necessary to improve their participation in decision making to enhance their empowerment as well as their status in the community. And thank you for listening. Thank you very much.
Okay, thank you, Doctor, for your presentation. Now we are moving to the last presenter with us, Dr. Delvin Al Ibrahim from the University of Duhok College of Science, Biology Department in Kurdistan region of Iraq. And she will present us the phenotypic study on multi-drug resistant ASBL producing E. coli isolated from the dairy farm. Please, Dr. Delvin, now you can start.
Thank you, Mr. Qais.
Okay,
I'm going to share the screen with you. I hope the screen is clear now.
Your presentation, and now you can start.
So good morning or good afternoon, everybody, according to the time in your country. It is my pleasure to present my work, a part of my PhD research in this form. So thank you for the conference committee to accept my presentation. So my research was under the title of Phenotypic and Genotypic Study on Multi-Drug Resistant ESBL Producing E. coli Isolated from a Dairy Farm. Due to the limitation of time, I'm going just to give a brief illustration for the subject background, some of the material and methods used, list some of the results, and going to the conclusion. So as all of you are aware that the antimicrobial resistance problem is a crucial problem. It is a worldwide agricultural and clinical problem which indicate the success and the separate speed spread of bacterial to adaptation to lethal selection in different environment. It is a crucial problem of great concern in public health, as they consider that food and food-producing animal, it's a potential route for the spread of such resistance antimicrobial genes and bacteria. E. coli as a commensal bacteria that can reside the intestinal of different animal can be used as a sentinel species for antimicrobial resistance in different reservoir. This bacteria can accept or acquire resistance very easily, which can be used as an early warning system to indicate the presence of antimicrobial resistance in even different other species. Here we can see the possible routes for the transfer of antibiotic resistance bacteria or genes between livestock, animal, and human. We can see that manure and slurry that is produced by livestock can work or be considered as a reservoir for antimicrobial gene and bacteria, and which can transfer these genes to the livestock itself or to the human being in different pathway, such as via the crops and soil. So what is beta-lactam? What is the beta-lactamase? Beta-lactam is the name of antibiotic group that have the beta-lactam rings within their chemical structure. This group of antibiotic can be classified to 4 main groups which include penicillin, cephalosporin, carbapenems, and monobactam, each which have a different chemical structure having the beta-lactam rings, and each group can be divided to different subgroups according to their generation of production. The mechanism action of this group of antibiotic is based on their activity on the cell wall synthesis of the bacteria. So specifically, they work on the transpeptidase step. The most common resistance of bacteria to resist this group of antibiotic is by producing enzyme to hydrolyze them. So these types of enzyme are called beta-lactamases. Beta-lactamases can be divided to extended spectrum beta-lactamases due to their broad spectrum of the substrate they work on, and also to the ampicillin types of beta-lactamases. Both of them can hydrolyze penicillins, third generation of cephalosporins, and monobactams, but there is some difference between both of them. I'm going to explain that more in the result discussion, in the result part. Other resistant mechanism also can be raised by bacteria, such as the reduced accumulation of the antibiotic and the action of the effluix pump. The main aims of this study was to determine the prevalence and the range of the presence of multi-drug resistant beta ESBLs, MMC beta-lactamase producing E. coli within the farm dairy, which is the site of the sample collection, to understand the diversity of resistance to beta-lactam antibiotic, and to make a link between the phenotypic profile for their resistance and the genotypic diversity in order to understand the diversity of the resistance, to determine the genetic genomic environment for the antimicrobial resistance